Hello everyone, my name is Julien Sescu and I am Head of Bioprocess Platform at TWB, Toulouse-wide Biotechnology. I would like to thank the IBISBA Organization Office to give me the opportunity to present the different services that TWB can offer. In this webinar, I will present you the TWB approach and tools concerning the setup of bioprocess from the early stages of a proof of concept to a 200-liter production demonstration. A typical situation that TWB often encounters with the contractualization with many biotech startups and with emerging projects of big biotech companies. TWB manages the whole workflow from strain engineering to 200 liters production demonstration. The different intermediate steps include genetic screening, phenotype screening, deep phenotype characteristic development, and optimization. The sequence or unit steps are applied on bacteria, yeast, or filamentous fungus. GMO or not GMO strain can be managed. The cell factories can be strict aerobic, facultative aerobic, or strict anaerobic. Final product can be the microbial biomass, protein, primary or secondary metabolites. Several steps can be managed in-house or perfectly integrated in a global IBISBA workflow, thanks to different IBISBA members, like Genoscope for strain adaptation by accelerated laboratory evolution, intensification by N2UA in Greece, or downstream processing by Vito in Belgium. For this webinar, I will focus my presentation on high-throughput screening and bioprocess optimization, two steps which are particularly determining in the course of the access to the market of a product. Bioprocesses involve biological catalysts that cannot be perfectly modeled numerically to provide reliable prediction of final performance. The choice and construction of the cell plant associated with its process requires numerous tests that must meet various challenges. For my part, I classify the challenge into three categories. The first challenge is to manage the huge biological diversity included in natural environment or generated by synthetic biology. The second challenge is to collect pertinent information in order to understand the key chemical, physical, and biological phenomena to be able to drive and increase global performances. The third challenge is to anticipate stress and constraints connected to final scale application at early stage of the development. The first challenge could be the management of biological diversity. Typically, for subjects dedicated to transfer a chemistry process to a biological process, a cell factory has to be built, and biological diversity is an opportunity. As a bench, this opportunity can turn into a nightmare if you have to determine which strain about 10,000 candidates will be the most efficient in 2 years in the process. The approach must allow choices to be made while minimizing the risk of choosing the wrong race horse. 3,200 liters can not be realized. The strategy consists of a succession of selection stages, progressively eliminating candidates who do not meet the performance criteria of the stage. Thus, the approach is to select the top 10% to 20% by gradually increasing the scale of implementation of the cell factory. At the beginning of the screens, the criteria are qualitative. For example, yes, no of the expression of one gene. The more the screening steps are linked, the more the criteria are quantitative. For the first screenings, high to very high throughput tools are used. For example, we can apply colony picker which can reach a screening rate of 3,000 clones per hour. Or cytometry sorter, we can reach a screening rate more than 20,000 clones per second. These first screenings allow to select hundreds of strains on growth or specific activity criteria. Following screenings in microplate or mini bioreactor context concern criteria of substrate compatibility, growth rate, pH compatibility, optimization compatibility, for example, compatibility with high substrate concentration, compatibility with fed batch mode, DSP compatibility, for example, secretion of the product, co-product accumulation. From 6 strains, bench bioreactor can be used in parallel for simultaneous process development. On finalist strain, evaluation for champion and vice champion. The late scale are used to validate the strain process pair in simulated industrial context. If champion pass all stress tests, a demonstration test can be realized. During this continuous and integrated workflow, there is progressive transition from screening to USP development to a final demonstration. The second challenge is how to measure to understand and to improve. Today, omics analyses are the preferred quantification tools of researchers to measure the living, to discover the mechanism underlying the biological functioning. Bioprocess developers have the same tools, but for more applied purpose. The analyses have to be applied with a permanent link to the bioprocess focus. Moreover, bioprocess developer has to find the most performant strain process pair for a specific application. Bioprocess developers have to detect for each step the main bottleneck. The main bottleneck can be located on the chassis, the engineered pathway, or in the process configuration. That is why, during the whole process development, the monitoring of the health of the strain bioprocess is mandatory at process efficacy level, global cell fitness, and metabolic pathway efficacy. Data must be collected in order to quantify synthesis of these 3 indicators. This is possible by testing the strain process under different modalities and by monitoring key variables. When number of strains is lower than 24, at TWB we use a custom platform, a robotic bioreactor platform, in order to test strain and bioprocess pairs, with data collection about bioprocess variables, for example, temperature control, pH control, oxygen transfer, the strain fitness, growth rate, viability, respiration rate, and with different process conditions, and with sample collection for metabolic pathway health evaluation. This platform integrates 24 reactors of 50 ml with conventional probe and 4 associated pumps for pH control and fed batch mode. By combining design of experiments and parallelized culture and online, at-line, and offline analysis, data exploitation allows to monitor bioprocess in bioreactor context in order to understand bottleneck in order to rapidly improve strain on our process. The third challenge is anticipate for derisking. Challenges around biotechnology are to switch from production biochemistry process with many steps to a one-step production by a microbial cell which catalyze, for example, 20 reactions from an industrial substrate within the cheapest and most profitable process. A molecular biologist will say you that the BL21 E. coli is really easy to modify genetically, and the fermentation engineer will say you that his stirring tank reactor accepts only strains which survives at pH 3 and no use amino acid from corn stip liquor. In order to benefit from molecular, microbiology and bioprocess level, all limits and possibilities of these three domains must be taken account. For example, fermentation team can advise the molecular biologist to choose genome integration rather than the use of plasmid to avoid antibiotic waste reprocessing cost. Molecular biologists can improve downstream process performance by deleting coproduct pathways. It is all about dialogue and open-mindedness. That is why the best way is to take account at the most early stage industrial constraints and possibilities, like to avoid crash during the first lab to pilot transfer. The industrial constraints can be classified in different categories, like industrial stress for microbial cell, legal constraints, industrial practices, or limitation by industrial infrastructures. The way to encompass industrial constraints at low scale can be well scheduled. For example, for culture medium development, a complexification of the medium followed by a simplification can be applied. Complexification in order to provide all unit nutrients for optimal growth or metabolic production with 4 different feed sources, for example, to manage specific unit nutrient consumption rates to evaluate nutrition needs. But after, simplification can appear by choosing industrial raw material and its treatment, which meet these nutrition needs. In order to apply this strategy, it is possible to use different tools based on culture parallelization. It is possible to use response surface plan to cartograph process performances versus control performances, or evaluate production performance by limited process control performances, for example, by limiting KLA at lab scale experiments. So, I hope that I convince you to get access to our IBISBA infrastructures and capabilities, to get access to the automated culture process ACP platform, and to get support for bioprocess development. Our fermentation platform houses BSCOP high and medium throughput cell culture station and bioreactor up to 200 liters and is operated by a dedicated technical research team. The fermentation platform is closely connected to an analytical facility, allowing to perform a large set of online and offline analyses in order to monitor fermentation processes. Thank you very much for your attention.